So while we're working on that, I'll go ahead and introduce the next speaker. So our second speaker will be Holly Heimsath. She works with Dr. Sally Permar on immune responses in neonatal or infant macaques. And she'll be talking today about her results on the correlations between the microbiome and immune response to HIV-1 vaccination of newborn Rhesus macaques. Holly?
Just a real quick note, if you would like to ask a question, I'll try and run this mic around so everyone can hear whenever you're ready.
Thank you. Great. Thank you, Kevin, and thank you to the CFAR for allowing me to share my work with you today. Our lab has a particular focus on pediatric HIV vaccination. Previous studies revealed infants can develop robust and durable antibody responses to HIV vaccination, similar to or of higher magnitude than adults who receive similar vaccines. If we can elicit protection during the critical window of the developing immune system, we hope it will provide lifelong immunity. Modeling studies suggest that a vaccine that protects prior to sexual debut would significantly reduce both adolescent and adult infections and therefore subsequent infant HIV infections. Vaccine effectiveness can be influenced by many factors, including nutrition, age, sex, and genetics. The intestinal resident microbiota are known symbionts for metabolism and for nutrition, and mounting evidence suggests a mutually beneficial relationship between the microbiota and the immune system as well. Establishment of the GI tract microbiota begins at birth and co-evolves with the immune system, dynamically influencing one another for the first 2 years of an infant's life, and it is within this window that we have the best chance of modulating the microbiome for increased vaccine response. Many groups have begun to explore the microbiome-immune system relationship. In one study, it was shown that human volunteers responding to the typhoid vaccine harbored more diverse, complex resident microbial communities. Another study demonstrated that probiotics augmented the antibody response to influenza, cholera, and polio vaccines, but not to DTaP, tetanus, or Hib. A third study showed that HIV vaccination induced GP41 reactive monoclonal antibodies that were frequently polyreactive with intestinal microbiota. These examples of studies relating the microbiota to the immune system are evidence that the microbiome impacts vaccination, but is likely specific to each vaccine type. Our goal of this preliminary study is to define the relationship between the developing infant Rhesus macaque microbiome and its immune response to HIV vaccination, and to determine how the infant microbiome can be modulated to improve response to HIV vaccination. Here is a schematic showing the immunization schedule for this study. Infant Rhesus macaques from 4 vaccine groups, conventional, co-administration, protein only, and extended interval, each consisting of 5 infant Rhesus monkeys, were immunized in varying HIV-OMV prime boost modalities and intervals, and by systemic and mucosal routes of immunization. The immunogen for all groups was the HIV-OMV protein GP120 from 1086C. The first 3 groups were immunized on a schedule of 0, 3, and 6 weeks of age, and the extended interval group was immunized at 0, 6, 12, and 32 weeks of age. For this study, we utilized stool samples collected at the birth time point and at weeks 8 and 14. We first asked how variable were the immune responses elicited by the HIV-OMV vaccine regimens. Measuring the immune responses, we tested the infant plasma by ELISA against GP120. Antibody responses peaked at 8 weeks for the conventional, co-administration, and protein-only groups, and week 14 for the extended interval group. These 2 time points coincide with 2 weeks after the 3rd immunization in all groups, and hereafter are referred to together as a peak immunogenicity time point. Using the binding antibody multiplex assay, or BAMA, we found that the peak plasma IgG levels to the C1, V2, V3 epitopes of GP120 did not differ between the groups. However, in the protein-only group, we saw a higher plasma IgG against the V1, V2 epitope when compared to the conventional group. We also measured plasma IgG antibody-dependent cell-mediated cytotoxicity activity against 1086C GP120 at the peak immunogenicity time point. All regimens induced similar titers of ADCC-mediating plasma antibodies. We found that all 4 vaccine groups induced neutralizing antibodies against the Tier 1 Clade C virus MW965 at the peak immune response time point. While there are appreciable differences in neutralizing titers between groups, we note that there are great variations within each group. We then asked, what is the microbiota diversity over time in vaccinated infant monkeys? We obtained our microbiota data via 16S rRNA sequencing. 16S ribosomal RNA codes for the part of the ribosome found in bacterial cells. Sections of this gene are conserved across genomes of all bacterial species, but variations between these regions are used to tell species apart and how they're related to one another. Utilizing the Duke Center for Genomic and Computational Biology, DNA was extracted from the stool, and PCR amplification was carried out using primers against the highly conserved section of the 16S rRNA gene. Variable regions were amplified and subsequently sequenced using the Illumina MiSeq platform. 16S rRNA reads were clustered into operational taxonomic units, or OTUs, and a subsequent diversity analysis was performed using the bioinformatics pipeline CHIME. This taxonomic summary shows the microbial diversity at the genre level over time and by vaccine group, with each bar representing an individual monkey. We can see that within vaccine groups, the diversity is highly variable, though there is an increased diversification as the infants age. So what factors best predict microbiota diversity? Principal component analysis of the 16S rRNA relative abundance data shows diversification of the gut microbiota clusters based on age. This age-based stratification is also seen when we apply a Shannon diversity index. When we break this PCA down by individual vaccine groups, we see that the same age stratification holds true for all groups. This phenomenon is not surprising, as it has been observed in both animals and in humans. We did look at other factors, such as birth location and age of the mother, to see if there were confounding factors, but nothing predicted better than age. We now wanted to know what is the relationship between the immune response and the developing microbiome. We performed Spearman correlations between peak immune response time points in the taxa at week 0. Bacterial taxa are along the y-axis, and the peak immune responses are along the x-axis. Peak immune responses include BAMA, neutralization, ELISA, avidity, and ADCC data. In this clustered heat map, the positive correlations are shown in shades of blue, and inverse correlations are shown in shades of red. The top 10 most positively correlated bacteria are boxed in blue. We can see that the correlations across the various immune responses are quite varied. We investigated this bacteria immune response relationship further by principal component analyses. In this PCA, we are asking if the taxa present at birth are predictive of peak antibody-dependent cell-mediated cytotoxicity against 1086C. Each circle is a monkey, and the color indicates the magnitude of ADCC activity, where blue is low ADCC and red is high. Low or high responders did not cluster together in any one region, leading us to conclude that week 0 microbiota did not predict ADCC activity. Conversely, monkeys with high neutralizing titers against MW965 and TZMBL cells are clustered together by principal component analysis. Using relative abundance of week 0 stool microbiota, we see that the animals with the highest neutralizing activity cluster together in the center of this plot. This was encouraging, as it seems that the bacteria present at birth are predictive of neutralizing titers. Now we wanted to know, are there specific microbes that drive the neutralization antibody response? We performed 2 further analyses to get at that question. Here, the random forest algorithm ordered by variable importance of relative abundance of the stool microbiota at birth, which predicted peak neutralization. We see proteobacteria shown in gray and firmicutes shown in orange are among the top candidate phyla. Interestingly, we see that 3 bacteria rise to the top as better predictors than environmental factors like vaccine group, birth location, and gender. We also used a regression analysis method called the least absolute shrinkage and selection operator, commonly called LASSO, which performs both variable selection and regularization in order to enhance the prediction accuracy and interpretability of the statistical model. And we're able to identify 3 candidate bacteria to explain the variation in neutralization titers. All 3 taxa belong to the phylum Firmicutes. Our results suggest that Doria and Enterococcus have positive correlations with high neutralization antibody titers, and Blautia has an inverse correlation with the high neutralization antibody titer. Together, these 3 bacteria are able to partially explain the peak neutralization titers. Looking back at the Spearman correlation heat map and the random forest algorithm, we see that Doria was a strong predictor of peak neutralization activity here as well. Since Doria has risen to the top of 3 separate analyses, it is our top candidate bacteria that we would like to pursue for further investigation. In summary, we found that bacteria diversity clusters according to age group, that variation in the peak tier 1 neutralization titers is partially explained by microbial communities found at birth, And we've identified multiple bacteria that have impact on aspects of the HIV vaccine response. And we believe that Doria, which appeared in 3 separate analyses, is a top candidate for continued exploration in determining its impact on the immune system. In future directions, we plan to perform a similar analysis of the oral microbiome. And we also hope to perform a rationally designed study administering a probiotic, perhaps Doria, alongside the GP120 vaccine to infant Rhesus macaques. We hope at the completion of the study, we will have identified a bacteria that will be used as a probiotic to increase specific and effective HIV vaccination response in infants, to yield lifelong immunity and bring us closer to an AIDS-free generation. And because nothing is accomplished alone, I would like to acknowledge our collaborators. I especially want to highlight Josh Daly, Erica Coons, and Dr. Justin Polera, who performed the immune-based assays shown today. Dr. Olaf Mueller, our bioinformatician, and Dr. Amir Ardashir, our collaborator at UC Davis, whose computational modeling and guidance has been essential in driving this project. I also must thank Dr. Sally Permar, my PI and mentor, whose level of dedication to this cause is inspiring. And last but certainly not least, thank you to our funding sources, without which this work would not be possible. Thank you for your attention. May I address any questions? We know that it is there. I don't know what the relative abundance compared to the infant microbiome is, but we do know that it is found in the human gut as well. And there are a couple of papers where we have seen an increased Doria population in humans will lead to a lower infection of C. jejuni, the bacteria that is one of the most common for causing food poisoning. There's also another study in humans that it marks that Doria is negatively associated with markers for insulin resistance. And then in a mouse study that there is one where we see that Doria also is negatively associated with inflammatory cytokines, IL-6 and MCP1. So I think that there is evidence that this really could be beneficial, and we would like to continue on that route to explore its potential in humans. All right. Thank you.
Can I ask one?
Sure.
So I was wondering, so have you actually taken the antibodies and shown if they can bind to this particular bacteria?
No, we have
So
not
do you
done
know
that
if
yet.
so could there be a causal link, or is it something else that you think is in there?
We are not sure, but that is something that we would like to do. We are talking with collaborators about isolating those bacteria and then running IPs and other binding assays to see if it is a direct or more of a systemic.
Okay.
So, welcome. Thank you.